Hi, I'm Tatiksha. Biochemistry is crucial in understanding the body's basic functions at a molecular level, such as digestion, energy derivation, metabolism, gene function, and immunological resistance. Medical biochemistry is essential for research in medical science branches.
Almost all enzymes are proteins, and they enhance rate of reactions. Enzymes do not participate in the reactions directly. Term enzyme was given by Kuhn. Buchner discovered or isolated zymase for the first time from yeast. Few nucleic acids also behave as enzymes, namely, ribozymes, ribonuclease P. Apart from above-mentioned RNA enzymes, all are proteinaceous in nature. There are two types of enzymes based on their structure. Simple enzymes, entirely made up of protein. For example, pepsin, trypsin, etc. Compound enzymes, these enzymes consist of two parts. Protein part, apoenzyme. Non-protein part, cofactor. Apoenzyme plus cofactor yields holoenzyme. Cofactors can be classified as under. Prosthetic group. This group has strong affinity for apoenzyme, that is, apoenzyme and prosthetic group are tightly bound to each other. For example, cytochrome, hemoglobin, myoglobin, catalase, and peroxidase contains heme group as prosthetic group. Coenzyme, it has transient linkage with apoenzyme. For example, NAD+ has vitamin niacin as coenzyme. Metal ions, many times, various metal ions are also seen as cofactors in enzymes. For example, iron is cofactor for catalase, chloride is cofactor for ptyalin, zinc is cofactor for carboxypeptidase. Factors affecting enzyme activity. Enzymes generally function in a narrow range of temperature and pH. Each enzyme shows its highest activity at a particular temperature and pH called the optimum temperature and optimum pH. Activity declines both below and above the optimum value. Low temperature preserves the enzyme in a temporarily inactive state, whereas high temperature destroys enzymatic activity because proteins are denatured by heat. With the increase in substrate concentration, the velocity of the enzymatic reaction rises at first. The reaction ultimately reaches a maximum velocity, which is not exceeded even by any further rise in concentration of the substrate. This is because the enzyme molecules are fewer than the substrate molecules, and after saturation of these molecules, there are no free enzyme molecules to bind with the additional substrate molecules. The substrate concentration at which the reaction attains half of its maximum velocity is equal to the Michaelis-Menten constant that is denoted by Km. The catalytic cycle of an enzyme action can be described in the following steps. First, the substrate binds to the active site of the enzyme, fitting into the active site. The binding of the substrate induces the enzyme to alter its shape, fitting more tightly around the substrate. The active site of the enzyme, now in close proximity of the substrate, breaks the chemical bonds of the substrate and the new enzyme product complex is formed. The enzyme releases the products of the reaction, and the free enzyme is ready to bind to another molecule of the substrate and run through the catalytic cycle once again. Transition state theory views chemical reactions as involving continuous changes in the positions and energies of atoms and molecules. It includes an activated complex state, known as the transition state, with maximum potential energy. The energy difference between the transition and initial states relates to the activation energy required for the reaction to occur. The graph given here shows activation energy in presence of enzyme and in absence of enzyme. Inhibition of enzyme action. Competitive inhibition. It is the inhibition of enzyme activity by presence of chemical that competes with the substrate for binding with active site of enzyme but does not undergo chemical change. This inhibitor chemical is also called substrate analog or competitive inhibitor. The affinity of the substrate for the enzyme is progressively decreased with the increase in concentration of inhibitor, lowering the rate of enzymatic action. Thus, the Km is high, but Vmax remains the same. Increase in substrate concentration reverses the effect of inhibitor. Examples: inhibition of alcohol dehydrogenase by ethanol in methanol poisoning. Inhibition by succinic dehydrogenase by malonate and oxaloacetate. Inhibition of para-amino benzoic acid, PABA, by sulfa drugs. Non-competitive inhibition. It is an inhibition of enzyme activity by the presence of a substance that has no structural similarity with the substrate. Here, the non-competitive inhibitor binds with active site or other than active site and completely inhibits enzymatic activity. Examples: cyanide inhibits activity of cytochrome oxidase. Diisopropyl fluorophosphate, DFPA nerve gas, prevents impulse transfer by irreversibly combining with serine of acetylcholinesterase. Allosteric modulation. This type of modulation is found in allosteric enzymes. The inhibitor is either non-competitive low molecular weight substance or product of a metabolic pathway having a chain of reactions involving number of enzymes. Thus, it is also called end product or feedback inhibition. Examples: inhibition of hexokinase by glucose 6-phosphate, inhibition of threonine deaminase by isoleucine. Km is not applicable to above-mentioned allosteric enzymes, because these enzymes are modulated by end products. Hence, substrate concentration, in this case, is not significant.